Thanks for joining us today for our monthly genomic epidemiology seminar. I'm really excited this morning that we were able to have Michigan come and talk to us. Our guest speaker was ill the last time she was scheduled, and now she is also out of office again. But Arianna Miles J was really gracious enough to step in and fill in her place. So I am going to go ahead and give her bio and introduce her, and then pass it off to her for this seminar. So Arianna Miles J, is the Genomics Analysis Unit Manager at the Michigan Department of Health and Human Services Bureau of Laboratories. She supervises a team of bioinformatics and genomic epidemiologists that implement all aspects of sequence data processing and analysis for a variety of bacterial, viral, and fungal pathogens at the Public Health Lab. And prior to joining governmental public health in around 2021, Arianna worked in genomic epidemiology in an academic research setting. She now works to apply this interdisciplinary training to expand the implementation of GenEpi within state and local public health through developing sustainable and flexible systems to process and analyze pathogen sequence data. So I'm really excited to have her here today. She is on the planning committee as well for the CSTE AMD workshop. And so we've got a lot of things in the hopper in terms of trying to, again, provide this applied GenEpi training and make it available for other state epidemiologists as well. So we're happy to have you today, Arianna, and I will turn it over to you.
Thanks, Jenny. Thanks everyone for having me. I was just telling Jenny, like, when I'm doing this presenter mode, I can't see chat or anything. So I asked Jenny to please or jump in. It's a small group. You can unmute and just interrupt me if anything is needed while I'm talking. And like Jenny said, I'm stepping in today to present sort of on behalf of Heather. So it's a little last minute, so bear with me if it's not as smooth as totally I would prefer. As well as I wanted to acknowledge that a lot of these slides, for especially the HAI-AR case studies, were prepared by Sarah McNamara, who's our HAI-AR epi here in Michigan and is also a genomic epidemiologist. So I wanted to just start out by kind of level setting on the definition of genomic epidemiology. So people here probably know, but I, wanted to kind of enter through that place and give a little bit of my perspective on it. So this one is just from the CDC Genomic Epi Toolkit. Defining genomic epi as the use of pathogen genomic data to determine the distribution and spread of an infectious disease in a specified population and the application of this information to control health problems. And I always like to emphasize when looking at this definition that really it's the definition of infectious disease epidemiology with this phrase, the use of pathogen genomic data. And so I always emphasize where it comes to genomic epi, to me, it's really defined by the types of questions that are being asked, being epidemiological questions. And then you have that added layer of using genomic data to do it. That really makes it genomic epi to me. So as you can imagine, this can look a lot of different ways. And today we're going to talk about some examples of some of those applications that we've had here in Michigan. I also always like to add one more phrase to this definition to not only look at the distribution and spread, but also features of an infectious disease. I think this is an application that is sort of a little more specific to genomic epi due to the high resolution nature of the data that you generate with genomics. You can address some interesting questions looking for example, like antimicrobial resistance genetic determinants or associations with particular genetic features, strain types, things like that with certain concerning clinical outcomes of interest. And that is like a sort of bonus, I feel like, application that comes through in genomic epidemiology. So to kind of break down those different pieces in that definition, I think they can kind of track towards different applications that you do see. So when we talk about the distribution of an infectious disease, one way to look at this in a molecular genomic epi perspective is more that simply like strain and variant typing and looking at the prevalence of a particular strain, variant clade over time and space, and having that provide you more information about what's going on with that pathogen. There's also the more like fine scale analyses that we do that compare things like SNPs to each other, really high resolution genetic comparisons. And those can be used for a variety of reasons, but a common one is to really look at like outbreak investigations, cluster detection, and kind of this spread term in the definition. And we will talk about a couple of applications today that relate to that as well. And it's, I would say, one that people are generally most familiar with. And then this features one, I have like the image of the antibiotic or drug next to it, again, identifying potentially genetic profiles, genes, features that are associated with certain phenotypes of interest in the pathogen, so like drug resistance, or potentially different outcomes of interest. That could be a clinical outcome of interest, for example, for like severity of disease. And at the end of today, I'll talk a little bit about an example of some work that we do that falls into this category. And I'll say, like, these are not hard and fast lines between these. They all sort of like blend together in different ways, but it can be useful to sort of try to think about sort of the binning of the different applications when you're thinking about genomic epi. And then I'll also note that one of the things that's cool about genomics and genomic epi is that like the building blocks for all these different applications really are the same. So you're tend to be working from the same sequence data that's being generated. You can use like varying types of epi data to go with it, but it really comes down to how you're summarizing the information, how you're analyzing it, how you're looking at it. And you know, part of what's cool about genomics is that you can use like sort of the same data sets and come at them at different angles to address these different types of questions. And again, just wanted to emphasize that it's helpful to me to think of like with genomic epi in the kind of classic infectious disease epi triad of environment, host, and agent. We're really taking this magnifying glass to the agent and just getting sort of really high resolution information about the agent. And that's what allows us to kind of subdivide it into these different categories to kind of tell us something that might look like all one thing without genomics and show that it's not all one thing, to look at those specific features. So it really, I think, just kind of delves really deep into the agent part of the epi triad. Okay, so let's jump into some applications. The first one I'll touch on briefly is about Candida auris. And it gets sort of at that distribution application. So I decided to spare you from showing you histograms of COVID variants today, because that's sort of a classic example of looking at like strain prevalence over time and space. I'll talk a little bit about C. auris in Michigan today and how even just classifying pathogens this like strain or clade level can like reveal interesting dynamics about the spread. So in Michigan, we have a lot of Candida auris, and it's been here really in force since 2022, and the case numbers have continued to increase. This is, I think, as of February, maybe or January, the data, but over 1,400 cases, most of which are screening cases, and some of which are clinical cases. But if you know anything about C. auris, you know that it is not all one thing where it comes to looking at the agent. There are 4 main clades that have been identified in the United States. This figure here on the left uses some of the older terminology where it was named after where it came from, but the one on the right, it's kind of small to read, but it uses, now we call them clades 1, 2, 3, and 4. And they are very genetically divergent from each other and are thought to have originated in different parts of the world, even though they are thought to have emerged at a really kind of similar time period, which is one of the sort of scientific mysteries of C. auris. But again, C. auris, we know, is not all one thing, and genomics is a technique we use to identify what type of C. auris we are working with. So a little bit more granular picture, again, going back to just like the case level in Michigan, you can see, like most infectious diseases and emerging infections when they enter our state, kind of you see the highest case numbers often in the Detroit and Detroit metro area. And then, it kind of starts to radiate out from there over time. And so these are the counties so far, I think as of early this year, that it had any C. auris cases identified, with the bulk of them being in Detroit. And then this is an example of a microreact dashboard that just layers on that clade information. So where it comes to sort of the molecular epidemiology of C. auris in Michigan, we have 3 of the 4 clades represented here. We actually also have had a clade 2 isolate like way back in 2021 that we don't have the sequence of. So all of the clades that have been identified in the US have been identified in Michigan, which I'm told is sort of just a common pattern. In Michigan, we tend to get like a little bit of everything. So clade 4 is definitely the most common clade, but you can see at the bottom of this screenshot of this dashboard, kind of basically like an epicurve, but only considering the sequenced samples. And you can see that the clade 1 prevalence is seems to be increasing a little bit here over time. And when you kind of map back again to the map, previously there was a case map and you saw that really high prevalence in the Detroit area, which you can see in the map that the dark blue is mostly the clade 4s, whereas some of the other areas that have more recently had C. auris introduced have dominance of clade 1, or at least a higher prevalence of clade 1. So overall, even not by even looking at SNPs and phylogenetic trees, even just by summarizing this clade-based information, it's clear from the genomic data that there have been multiple introductions of C. auris into Michigan, not just a single introduction that has spread widely. And even within those clades, that seems to be likely that there have been multiple introductions even within the clades, especially in clade 4, where we have those higher numbers. So again, just kind of revealing that complexity and then adding this layer of distribution of different clades across the state. Of course, we can delve deeper into more SNP distances, phylogenetic trees, things like that, to get a little more granular within each clade, both at these questions of likely introductions, as well as trying to address certain questions about potential outbreaks or genomic linkages that are consistent with epilinkages. If anyone here has worked with C. auris genomics, you know that it can be a little trickier than some of the other pathogens we've worked with, as far as there's actually kind of a lot of genomic homogeneity. And so getting that resolution and kind of understanding the relationship between certain clusters and SNP distances and epilinks is just a less developed, less studied area than it is in some other pathogens. But it's definitely something we've been investigating for a while here in Michigan. Now I'm going to move on to another fungal example and talk a little bit about a blastomycosis outbreak that occurred here in Michigan. So there was a large outbreak of blastomycosis that occurred at Paper Mill on the Upper Peninsula, Michigan. And if you're interested, there is a publication recently published in Clinical Infectious Diseases about this. Although I did check this morning and I'm pretty sure it's behind a paywall. So even as a co-author, I couldn't open the final article. So there's that. But you can see in this graphical abstract and in reading the abstract, it was a large outbreak of blastomycosis, 131 cases. And it was associated with a paper mill in the Upper Peninsula of Michigan. And this paper is more of a clinical focus of the paper. They looked at patient outcomes and patient risk factors and kind of hypothesized some kind of factors that could lead to less poor outcomes in patients. But one piece of this paper, the Bureau of Labs was involved in identifying the pathogen that was in the outbreak. So this is the application that is, I guess, not strictly genomic epidemiology, just kind of like an identification, but certainly you need to have an identification before you can proceed with a lot of the genomic epi applications. So this is a figure and it demonstrates how we identified Blastomyces gilchristei as a species, which is up in the red at the top of the tree. The samples on the tree that have B and G are the outbreak isolates, and the ones with the GCA and GCF are contextual samples from public databases. So this is an example. Sometimes, like mycology or microbiology, can only get to a genus level or only get to like a complex level in their identification methods, whether it be microscopic or using MALDI-TOF or something like that. So sometimes sequencing can come in and give a little bit more of a specific diagnosis. And so that was, this was all part of surveillance, but this is what happened in this outbreak to narrow it down to be Blastomyces gilchristei rather than the slightly more common Blastomyces dermatitidis. And then beyond that, this is an example of an outbreak where, like, I think it's important to think about in these applications. Sometimes the epi is there, it's really strong, and the sequencing sort of confirms things. And then sometimes, which we'll talk about an example of this later, the sequencing reveals something that was maybe not immediately apparent based off the epi data that had been collected. And then you can kind of like go back and get more epi data that potentially confirm the linkages you're seeing. So this is an example of like, this was like a large outbreak. It was very clearly associated with this paper mill. And there, as you saw, there were 131 cases. We sequenced, I think, like 11 of them or something. It's not trivial to recover isolates associated with this outbreak. But it was one of those situations where everything we saw did make sense. The known outbreak associated samples did cluster together, which you can see at the top of this tree. The outbreak ones are blue, whereas the red ones are some contextual samples of gilchristei. And then there's a couple interesting samples in here too that are contextual from Michigan. One of them, that 23BG16, which you see just like a little bit outside of that main cluster, that was a sample collected from a patient also in the Upper Peninsula, but in a different region of the Upper Peninsula. So didn't have that paper mill association, and you can see that there's a much more genetic divergence there with that sample. The purple triangle that's nested in is also another interesting one. This was a sample collected from a dog that was near the paper mill, that resided near the paper mill. And it clustered in pretty closely with the outbreak isolates. So, it's one sample. There's a lot of different hypotheses that could connect the two. But, this is an environmental pathogen. And so it highlights some of that One Health perspective of our companion animals can also be at risk for some of this stuff. So this was, again, an example where the sequencing data was used to really help with that identification and then confirm the clustering that was observed based off of the epi data as well. Okay, so those are my couple fungal examples, and then I will also provide a couple of bacterial examples. The viruses are neglected here in this talk today. But my next one is going to be talking about carbapenemase producing organisms and how routine genomic surveillance picked up a couple of endoscope associated outbreaks here in Michigan. So carbapenemase producing organisms, there's a surveillance program in Michigan. It's a reportable condition, and cases are submitted to our disease surveillance system. It includes Enterobacteriales as well as Pseudomonas aeruginosa and Acinetobacter species. And isolates are forwarded to BOL for additional characterization, including whole genome sequencing for a subset of those, which I will describe a little bit more detail in the next section. So the isolates enter through the clinical microbiology lab. They undergo the initial organism ID confirmation via MALDI, and then the phenotypic testing and genotypic testing for presence of carbapenemase enzymes or carbapenemase genes, as well as some antibiotic susceptibility testing. And then basically the KPC producing organisms are not routinely reflexed for sequencing because there are so many of them, they're so prevalent, but the non-KPCs, so your IMS, BIMs, NDMs, are reflexed for whole genome sequencing, as well as some like pan non-susceptible isolates. There's a couple of other categories, but other than the KPCs, we sequence a large proportion of them that are positive for these carbapenemase enzymes by some of these other testing methods. And then once they're reflexed for sequencing, they obviously go through the sequencing process of the nucleic acid extraction, library prep, and sequencing. They go through bioinformatic analysis, which happens in our area. We use the CDC's Phoenix pipeline to do the initial QC, taxonomic identification, AR gene detection processes. And then we have a custom pipeline that we do in-house for the clustering type analyses, which has evolved a lot over the years as we've the volumes have increased. We've kind of had to use different criteria, both to group things together to analyze them, as well as to determine when we should generate reports. So when reports are generated, they do get shared with our antimicrobial resistance epidemiologists. And then we also have some dashboards that keep like higher level a view of things, of the strains that are circulating within each species. So this is always going on in the background, and this is a couple stories of some interesting outbreaks associated with endoscopes that were revealed through this work. So this first one was back a little after I started in 2021, was an example of two genomically linked cases from two patients that were E. coli ST648 and had an NDM5 gene identified. And they were genetically identical through the analysis that was in place at the time of 0 SNP differences. So this information made it to the AR epidemiologist who started to dig into what's going on with these patients. Patient one was admitted from home to hospital A in June of 2021, had a liver transplant due to history of liver cirrhosis, and had a lengthy hospitalization where they developed a biliary leak and had ERCP procedure with a scope, and then subsequently had exploratory surgery. They had an intra-op culture that grew an NDM E. coli. During the initial investigation of this case, there were no other known NDM positive close contacts. Patient two was admitted from home to a different hospital, Hospital B, in August of 2021, a couple months later. They had a fall from home after chemo treatment for a metastatic colorectal cancer and developed septic shock. Had blood cultures that grew an NDM positive E. coli, and unfortunately, the patient did expire in this case. So during the initial investigation of this one, again, there were no other NDM positive close contacts. This was this patient's first encounter at that hospital system, but then they were lost to follow-up, obviously, when they expired. So after whole genome sequencing, this was an example where it led the EPIs to dig deeper into potentially if there could be any links and talk to these various facilities. And when they did that, they discovered that patient B actually had been previously admitted to Hospital A in July of 2021 and had had a scope-based procedure for tumor-related obstruction. So they actually determined that both patients had had their ERCP procedures on the same day with the same duodenoscope. There's a couple different types of scopes to understand. There's this duodenoscope that goes into this ERCP procedure, which is used to diagnose problems in liver, gallbladder, bile ducts, and pancreas. And then there are also gastroscopes that are used to diagnose problems in the upper gastrointestinal tract. And both types of scopes are definitely a risk for pathogen transmission. They have factors that contribute to their contamination can include they're really intricately designed and difficult to clean and disinfect properly. They can have internal defects that are difficult to detect. Reprocessing them can be a complex process. Of course, the healthcare environment can be contaminated. There can be issues with staff turnover and adequacy of training in the cleaning process. And there can be really rapid turnover of scopes between these procedures. Procedures can happen at a pretty high volume. So in this case, there was continued surveillance, genomic surveillance that did eventually ultimately detect 9 cases with the NDM E. coli. And these cases all clustered together pretty closely within the like 25 SNP range. So this was type of information that we provided as we continued to support the ongoing investigation. And then, this is one that kind of flips back and forth between the genomics and the epi. So the epi, when you actually narrowed it down to those 9 cases, revealed that there were multiple scopes that were involved. So it wasn't just a single scope used between all of these patients, but multiple different ones were involved. So they were able to, through identification and narrowing down the cases with the genomic data, really track down in detail which scopes they needed to remove from circulation. So that was like part 1 of one scope-related outbreak. A couple years later, there was a kind of a similar initial start to this sort of situation where there were 2 cases with 5 SNP differences, also NDM5s. This one was E. coli ST167. This is a different facility, different time. In this one, they started to dig into it. Patient 1 was admitted from home to Hospital A. In July 23, they had a small bowel obstruction and had exploratory surgery. Blood culture grew NDM E. coli and then subsequently had an EGD scope procedure done at the bedside. Patient 2 was admitted also to Hospital B, again, different hospital, in August 23. They had physical rehab following recent hospitalization for complications from lung cancer and had a urine culture that grew the NDM E. coli. But they had previously been admitted to Hospital A in July and August of 23 and had also had the EGD procedure. So for both of these, there were no known NDM positive close contacts that were otherwise identified, and no other significant epi links besides the shared gastroscope of this procedure. So in this one, fortunately, there were just the 2 genomically linked cases. They were about 5 SNPs apart and definitely clustered separately from the other contextual isolates of sequences we had here in Michigan of that same strain. And in this one, it was the one gastroscope A, that was identified as the culprit of the shared link between both patients. So important to note that both of these outbreaks resulted in extensive infection prevention and control follow-up. They pulled and inspected the scopes. In both cases, I believe they didn't identify lapses in reprocessing internally, but there was some internal damage noted in some cases. They were not able to recover any positive cultures from the scopes themselves. Again, they continued to do the surveillance in outbreak 1. They did identify 7 additional cases beyond the first 2, and in outbreak 2, there were no additional cases identified. Outbreak 1 had more contact tracing. They had more than 1,200 patients that were exposed and 200 screened, but all were negative. In the second outbreak, 118 were exposed, 80 screened, but were all negative. And then the different approaches they took in the first outbreak, they replaced the entire fleet of scopes to a newer design that had disposable end caps. In the second outbreak, they switched to fully disposable scopes for known MDRO positive patients. Both facilities implemented additional QA steps to the scope reprocessing as well. So that is an example of a, again, more of that like genomics first situation where you have that surveillance kind of ongoing in the background, and the genomics helps identify something that maybe identified sooner than would have necessarily been identified with the epi alone, especially given the rising prevalence of these NDM producing organisms. Unfortunately, it can be messier to figure out what is linked to what. Okay, so I'm going to end with slightly different application to just touch on that features part of the definition that I added and talked about, which is really delving into the genomic data to look for features of interest clinically in this case. So we have some work going on to investigate genetic determinants of Estronium avibactam resistance in carbapenemase producing organisms. So for those that aren't familiar, Estronium avibactam is just one of the newer drugs that are available to treat carbapenemase producing organisms. When I was looking for this information, I think it was only recently fully approved by the FDA. Because it's newer, that also means that they haven't yet identified cut points to determine non-susceptibility and resistance where it comes to the antimicrobial susceptibility testing that they do. So that's something that's like a little bit more up in the air. It's also less common to have susceptibility testing, antimicrobial susceptibility testing capabilities in laboratories because it's a newer drug and newer types of tests that they'd be doing. But despite it being new and sort of like a harder hitting one for those difficult to treat situations, there definitely already is reduced susceptibility to this drug. And you can see that reflected in the genomes of the organisms as well. So there's been some research that has looked for associations, genetic associations between certain resistance determinants, potential resistance determinants, and especially combinations of them, and then phenotypic reduced susceptibility to these drugs. And so this is an example where, because the antimicrobial susceptibility testing for this drug is not widely available because it's a newer drug, because they don't have the cut points available, things like that. It's an example where the genomic data potentially could help supplement providing a picture for pharmacists and providers to the prevalence of the resistance determinants in the state or in different regions could kind of potentially link to the prevalence of resistance and could inform empiric choices for antibiotic treatment for patients. So we have been looking at this in some of our data. So there's a particular mutation like in the NDL, like metallo-beta-lactamase producing E. coli in particular, a lot of NDMs. There's this PB3 mutation that has been in previous research associated to be not a sufficient, but a part of the contributing factor to resistance in E. coli to this drug. And so it's something like working in public health and having these isolates from across the state, we can look at the distribution of that in the data that we have available, including across different sequence types or strain types. And you can see that it's not necessarily equally distributed across different strains of E. coli. And then we're also in the process of working with our microbiology colleagues to collect more of the antimicrobial susceptibility data to potentially look more closely in our own setting at those correlations between different gene presence, absence, and resistance that we see. And this is a question that's of a lot of interest to our kind of pharmacy stewardship leaders in the state, because it's like a situation where they're sort of flying blind. They don't know how much resistance might be out there or where it might be located. So to the extent that the surveillance data that we collect for all these other purposes can be repurposed as well to provide insight into some of these questions, that would have a public health and clinical benefit. So this is an example of kind of that features type question to look for those associations between certain genes and certain characteristics of interest. And I think this one, like, in some ways you look at it, can be epidemiology, especially if you're looking at the distribution and looking at it like as a risk factor for clinical outcomes. Some angles you look at it, maybe you wouldn't call it epidemiology, but it's definitely of public health and clinical interest. Okay, so that was just a little a whirlwind tour of sorts through some applications of recent applications of genomic epi here in Michigan. And again, like, I think a cool thing about genomic epi is that you can use these same types of data and analyze it in different ways to really get at different applications depending on what your needs are. Describing distributions, investigating outbreaks, and examining features of interest or concern are all applications that do occur in public health. And in Michigan, we're doing a little bit of all of these across a variety of different pathogen types. So that is all I have. I'm happy to take questions. And again, thanks for inviting me to share today.